Hello, I'm Bill Griffiths from Swansea University, and I'm the host of today's Lipid Maps webinar. So first of all, thanks everybody for joining today's webinar, which will be presented by our guest speaker, Marc Poirot from Toulouse. The title of Marc's talk will be Recent Discoveries on the Cholesterol Metabolic Pathway, the 5,6-epoxy cholesterol branch, dendrogenins and oncosterone. So some housekeeping details before we start. The webinar is being recorded and will be uploaded onto the Lipid Maps website and YouTube channel within the next 48 hours. The webinar will be around 45 minutes with 10 minutes at the end for questions. And please use the questions and answers function throughout the presentation to ask your questions. And at the end of the seminar, we'll unmute you to ask your questions directly to Marc. Today's seminar is being sponsored by Cayman Chemical, and we're very grateful for their ongoing support. So finally, just before Marc starts, a few words about Marc. So Marc studied organic chemistry and biochemistry at the University of Paul Sabatier at Toulouse, where he received his PhD in chemical biology. He then did a postdoc at Sanofi Elf Bioresearch and was then a visiting scientist at NIH working on steroid hormones. Marc is a group leader with his longtime collaborator Sandrine Sylvain-Poirot at the Research Centre in Toulouse. His interests include cholesterol metabolism and its dysregulation in cancer. Marc's published more than 130 papers, and he will receive the 2023 Schroefer Medal awarded by the American Oil Chemists Society in recognition of his work in the field of sterols in the next month or so. So Marc, the floor is yours. Please share your screen and off you go.
Okay, thank you so much, Bill, for your kind introduction and for your invitation to present the work of my team. This is a great honor to be with you at Lipid Maps. And so I'm going to talk about what we found recently, but from the beginning, actually. So actually, what we have done is we were working on tamoxifen, and tamoxifen is a great drug, one of the first targeted therapy imagined by Greg Jordan in the 70s, and which is one of the first drug approved by the FDA for the prevention of breast cancer. So it's really important to understand perfectly how it works. So people know that these compounds have been conceived to block the mitogenic action of estradiol, which is a tumor promoter on breast cancer. And my PhD supervisor, Jean-Charles Fay, who passed away unfortunately in the fall of last year, identify or characterize a high affinity binding site named anti-estrogen binding site involved supposedly on the resistance to tamoxifen and on cytotoxicity of these compounds. And I did my PhD with him and developed a specific tool to characterize this binding site. And what we found actually is that this binding site was made of two cholesterogenic enzymes, the sterol isomerase and the dehydrocholesterol reductase, the 7-dehydrocholesterol reductase. And what's going on when tamoxifen interact with this binding site it induced the accumulation of a sterol named zymostenol. And this was interesting because Jilling and Mietenen's group showed in the late 90s that a patient treated with tamoxifen accumulate zymostenol in their plasma. So we studied that in vitro and found that actually the accumulation of these sterols was associated to the induction of autophagic process. This is autophagosomes that appears in cells treated with tamoxifen or with the selective ABS ligand that does not interact with estrogen receptor. That can be seen very easily with Philippine labeling, the accumulation of these sterols, and appears as multilamellar bodies by transmission electron microscopy. Actually, what we knew also is that the ABS was known as an oxysterol binding site. This is well reported by Huang and Martin in their J-lipid research paper. And the ABS was present in the microsomes, and one inhibitor, the 6-keto-cholestanol, was found to be the most potent endogenous inhibitor. And this was quite similar to what was described by Sevanian for an enzyme named as the cholesterol 5,6 epoxide hydrolase. And actually, we showed and published that in PNAS that this ABS carried out the CHH activity, which means and the isomerase was involved in the displays the catalytic site, while the other enzyme was more involved in the regulation of this activity. So that's what the starting point for us to be involved in cholesterol epoxide metabolism. Sorry, and the tamoxifen and PBP inhibit strongly this enzyme at pharmacological concentration. And we found that these epoxides were actually involved in the very interesting process such as redifferentiation of breast cancer cells as well as cytotoxicity. So this was interesting, but also quite alarming because 5,6 epoxide of cholesterol were proposed to be carcinogenic by Sevanian's group in the early 80s. And nobody was known about the potency of that kind of compound to be an alkylating substance or a direct carcinogen. So this is something we check in the lab. You know, styrene oxide is an epoxide that is very sensitive to nucleophilic substance and get conjugation spontaneously in their presence. And this is a typical compound that is an alkylating or mutagenic substance, but it's classified as a probable human carcinogen because it was not really clear using in vivo experiments to demonstrate that this compound was a mutagenic or carcinogenic substance. What about 5,6 beta epoxide of cholesterol? It is totally unreactive in the presence of nucleophilic substance, even in the presence of a catalyst such as Lewis acid or heat. And so that rule out the fact that it could be an alkylating substance. The alpha epoxide of cholesterol, which is the other isomer, is not reactive at ambient temperature, even at 37 degrees. It does not react with nucleophilic, but in the presence of a catalyst such as Lewis acid or heat, it can give a conjugate. And what was interesting is that we got a single isomer among 8 different possibilities. And this suggests to us that it could represent a possibility of a metabolic pathway if that kind of transformation exists in mammalian tissue. And if you look now at the cholesterol metabolism pathway, you know, the two subunits of the CHH are carried out by two enzymes that for which null mutation are associated with severe genetic syndromes such as Smith-Lemmi-Opitz syndrome or Conradi-Unermann. And we postulated that 5,6-EC or 5,6 metabolites, if they exist, could contribute to the control of cell differentiation. And we did our first experiment where to produce conjugates with histamine or polyamines, and we obtained a single product of conjugation. And these two of these compounds were very interesting because they induce at low concentration, nanomolar concentration, they induce differentiation of different kind of cells in vitro. And we observed two different comportments. With the conjugate with histamine, we got cytotoxicity, while with the conjugate with spermidine, we got no cytotoxicity, but we found that it seems that these compounds at 1 micromolar at least was selective of some cell subtypes. And we found, for example, that we can induce the differentiation of glioma or neuroblastoma into neuron, of melanoma into melanocyte, but just with DDA, and of monocyte into dendritic cells after pretreatment with a 4-bol-ester. And we call this compound dendrogenin because it induces dendrogenesis on these cells. And we found later that we can also induce cell differentiation of breast cancer into breast lactating cells, or also thyroid carcinoma cells into redifferentiated thyroid cells that reactivate, in which the iodine metabolism is reactivated. What's going on if you inject that kind of compound at low doses into a tumor implanted in mice? You can observe a strong decrease in the development of the tumor, while the geometric isomer was ineffective, actually. And tamoxifen for that kind of breast cancer cell lines required a high amount in compounds. And we got a nice survival effect with that kind of compounds. What we observed in tumors is that we activate the presence of marker of differentiation in these cells, such as the accumulation of triglyceride and the presence of a protein that can be found in milk during lactation. Next question was, is it a metabolite? So since we have synthesized these compounds, we have been able to set up an analytical methods, and then we analyze extracts from mice and found actually at the retention time corresponding to synthetic DDA, we found that the presence of compounds with the mass, not only the molecular mass, but also the fragmentation that correspond to authentic DDA that demonstrate the existence of this compound as a metabolite in mammals. We found also that the incubation of precursors with histamine and 5,6-epoxide of cholesterol enable the production of the DDA, which does not appear in the presence of the beta epoxide. and that disappeared when we boiled the extract or treat with a protease, showing that this reaction was done with a proteinaceous enzyme. Next, we check if that kind of compounds can be present in a number of different kind of cancer cell lines, and didn't find the detectable amount of DDA, except on normal mammary epithelial cells. And when we check biopsies from patients with breast cancer, we found that when we compared the level of DDA, first we found DDA in the breast from patients, and when we compare normal adjacent tissue to tumors, we found that a strong decrease below the active pharmacological active concentration of DDA in tumors, evidencing a deregulation in DDA metabolism in that case. That could reflect a biosynthesis defect, the absence of enzyme or a catabolism. This is something that we did progress on and will publish soon. What is the efficacy of such compound on human cancer cells, and what is the mechanism of action? So I will shorten this aspect, but we were focusing on elixir. Elixir are nuclear receptors that are ligand-dependent transcription factors that act mainly as co-repressor of a number of genes. And actually, we demonstrate that that DDA can antagonize the 22R hydroxycholesterol and compete directly on the ligand binding domain of these receptors, showing that DDA was a ligand and a modulator of elixir isoforms. And when we did some docking experiments on the crystal structure of elixir beta, this is the example. What was interesting is that the different group present on DDA give high energy interaction with residues present on the ligand binding domain, showing that actually that kind of compound was made to interact with elixir. Looking at the effect of compounds on different kind of cancer cells, this is melanoma cells, mouse and human. You can see that DDA induced the accumulation of acidic vesicles that can be labeled by monodontyl cadaverine, and activate the expression of LC32. This is ATG8, and stimulate an autophagic flux within this cell. And this effect was elixir dependent, because when we did knockdown of elixir, we lose part of this effect, cytotoxic effect, and showed also that autophagy in that case was actually the main mechanism associated with cell death induced by DDA. We do not observe any toxic effect with agonists, but these elixir agonists were able to inhibit part of the effect induced by DDA on these cell lines. And in vivo, the effect was similar. That means that the presence of elixir was required to induce a control of tumor growth for human and mouse cells implanted in mice. And we got the characteristics we observed in vitro. And that means that according the concentration with DDA, we can induce cell differentiation and growth arrest, and then after lethal autophagy that is associated with cell death. We found the mechanism, and what was interesting is that we observed the appearance of differentiation markers. We found also that DDA activate the transcription of a number of lipid biosynthesis enzyme, and on the other way, we activate the expression of master regulators of autophagy and lysosome biogenesis. And also, when we treated cells on a syngenic situation on normal mice, we found that DDA induced the infiltration of T lymphocyte and dendritic cells, suggesting that the anti-tumor effect of DDA could be associated with the activation of the immune system. And this was not observed when we implanted such tumors on immunodepleted mice. So to look at that, then we check what can be produced by tumor cells treated by DDA. And one of the first experiments we did was to check the ultrastructure of cells treated by DDA. And you can see here, you have multivesicular bodies and grapes of vesicles that suggest that the compound induce the production of exosomes or small extracellular vesicles. And when you treat cells at non-toxic concentration with DDA, this is the example of BC, you can see a strong morphological changes. And also we found that we induce dose-dependent production of these vesicles. And when we take these vesicles and inject these vesicles in tumors in which we have implanted tumor cells, then we saw a very nice anti-tumor effect, showing that these vesicles display anti-tumor properties. And actually, we did just 2 injections with this extracellular vesicle, and the effect was quite impressive. What we found of interest is that DDA stimulates the production of very peculiar lipids, which is LBPA or BMP. And this effect was elixir dependent because we lose this stimulation of production of compounds when we did knockdown of elixir beta. And also other agonists such as 22R hydroxycholesterol inhibit the basal production of BMP and also the stimulation induced by DDA, showing that the effect was controlled by elixir beta. And to summarize the effect of DDA on cancer cells, it depends on what kind of concentration you use, but we found elixir-dependent events at high concentration that induce lethal autophagy, and at lower doses, we induce the production characteristics of differentiation on cancer cells and the production of vesicles that activate TC maturation and T-cell activation. And this effect contribute to the anti-tumor effect of the compound, suggesting that probably the endogenous function of DDA could be the immunosurveillance of tissues. And so we can add now that associated with cell differentiation, we produce anti-tumor small extracellular vesicles that contribute to anti-tumor effect of DDA. Now, we have seen that DDA is not produced in cancer cell, but what is the metabolism of 5,6-epoxide of cholesterol? If you incubate cells with labeled 5,6-epoxide of cholesterol, you can compare the metabolism. This is normal cells and cancer cells. And we found another metabolite here that appears. And Philippe de Medina characterized it as the 6-oxocholestanediol. So BC cells can transform very strongly cholestanediol into 6-oxocholestanediol, and these compounds display tumor promoter properties in vitro. It stimulates the proliferations of different kind of of breast cancer cells. It stimulates also breast cancer invasiveness, and in vivo stimulate the proliferation. And we don't need, like for 27-hydroxycholesterol, to encapsulate it in cyclodextrin. We just have to inject that kind of compounds to observe this effect. So that means that on this pathway, we can act the production of oncosterone, and we identify the enzymes involved in the production of oncosterone. And on the reverse reaction, these are the 11 hydroxysteroid dehydrogenase of type 2 to produce the oxidation, and the reverse reaction was catalyzed by the 11 hydroxysteroid dehydrogenase of type 1, but in the presence of glucose 6 dehydrogenase. And we also characterize the mechanism involved in the effect of oncosterone, which involve glucocorticoid receptors that is mainly involved in the control of cancer cell proliferation. And Elixir is also another binding receptor for oncosterone, and it seems to be more involved in cancer cell invasiveness. So if we have shown that 5,6-epoxide of cholesterol are not alkylating substances, they are meanwhile controlling carcinogenesis. So we have identified this pathway on a new branch on the cholesterol pathway that involved 5,6-epoxides, and the alpha was shown to be transformed into dendrogenin A that display very interesting properties and behave like an oncosuppressor metabolite. This is really a very interesting properties. And on the other hand, 5,6-epoxide can be metabolized by two enzymes, a cholesterol epoxide hydrolase that we characterize, and with HSD2, it can produce oncosterone, which stimulate proliferation and invasiveness of cells. And so what we found is that by studying tamoxifen, we could identify this enzyme as a target for these compounds, which was involved in the production of very interesting sterols, and give us the idea to look at possible transformation of epoxide, which make us identify dendrogenin A. Next, we are very interested in looking at if a specific enzyme can produce the 2-diastereoisomer of epoxide. We know that lipoperoxidation, for example, can produce both isomer, and probably under the context, membranous context, we can have more of one or the other. We are interested in looking at the metabolism of these compounds to determine whether they are end products or not. And now we are looking if other conjugates could exist also. So that work is currently done by several people in the lab. Obviously, my co-PI, Sandrine, is at the origin of most concepts observed by the team. And we are very proud of being able to look at that. And I thank all the sponsors and all my collaborators that contribute to this project since several years now. So thank you for your attention. I'd be very happy to answer your question if you have some.
Okay, thanks very much, Marc. I guess we've all learned a lot there about new pathways of cholesterol metabolism. So Marc will take questions. So if you put your questions into the Q&A, we can then unmute you, and you can ask them live. So I think Jean-Marc, you might have had a couple of questions there in the chat. So Jean-Marc, would you like to ask your questions?
Can you hear me?
Yep.
Yeah, yes.
Jean-Marc.
Thank you. Hi, Bill, and salut, Marc. Okay, thank you, Marc. It was really an interesting and ecstatic work you've done since many years. I have a question regarding the extravesical. You didn't say, or maybe I missed something, what could be inside. Is it some oxysterols or some siRNA, some miRNA, something like that?
They contain a lot of things, obviously. We haven't published all they can be present in this, but what we found is that the treatment with DDA stimulate the production of proteins involved in antigen presentation. You know, and this is a key parameter that make these vesicles activate the immune system against the tumor.
Okay, really interesting. I have maybe a second question, Bill. May I ask the second question?
Yep, go on.
Yeah, and the second question regards oncosterone. You didn't say whether oncosterone was acting as an agonist, an antagonist, a reverse agonist of elixir beta. Does oncosterone has the same effect on elixir alpha? Have you done all the work on this compound and elixir beta together?
This is something that is ongoing on the lab. What we found actually is that oncosterone is probably a biased agonist. You know, we have seen that we can activate a number of genes under GR control, but not all that are activated, for example, or repressed by cortisol, for example. So this is something we are working on because this is very peculiar to that kind of compound, and probably that means that there are some crosstalk between a differential crosstalk between in terms of gene activation between steroids and sterols on that point. And actually, this is very intriguing and also very interesting. Now we try to do some structure function studies on that kind of sterols to determine more precisely what can control their specific capacity to control the mitogenic pathway, for example. And this is something that is done by a PhD student in our lab right now.
Thank you, Marc.
You're welcome.
Okay, I see Dieter John is there. Dieter, please ask your question.
Yeah, thank you for an interesting talk. I just came back. Last year, I listened to the Nobel lectures, and I listened to Carl Sharpless, and I learned during my studies in organic chemistry the epoxidation of allylic alcohols, and cholesterol epoxidation looks like a cyclic allylic epoxidation, and that was called Sharpless oxidation, and now he got the second for click cholesterol. But could this help the synthesis of this epoxide, help us to find this natural epoxidation process that you described at the end?
I'm not sure to have well understood what you say. You mean that?
So the formation of epoxide from cholesterol. And there's on the 3 beta position, there is a hydroxy group. So it is an allylic alcohol. And Sharpless made this epoxidation of normal allylics. So it was just an idea. Perhaps I will just make a painting and send it to you. Perhaps then it's more clear to understand.
Yeah. Actually, yes, you can consider different way to look at that. I think that in terms of autooxidation, this is something that is quite clear and have been developed by a number of researchers. Even recently, they published a very interesting paper in JACS on that aspect. But what I think is that if there's a metabolic pathway generating based on the stereochemical, stereoselective production of compounds, it might exist an enzyme that produce stereoselectively the alpha epoxides that can drive this pathway. And this have been described in the late 70s by Watabe's group in Japan, but never characterized precisely. So I am pretty sure that we will find some. We have preliminary data that seems to indicate that a specific P450 could be involved in either the production of the beta epoxide or the alpha epoxide. And this is something we are working on.
But then you presume that it's a cytochrome P450. Then you can do the reaction with and without NADPH or NADH.
Yeah, sure.
Yeah. Thanks.
You're welcome, Dieter.
Okay, I see Ernest is here. Ernest Arenas, please ask your question to Marc.
Hi, Marc. It's Ernest here. Very nice talk. I was wondering if DDA is also capable of blocking the effects of 24-25 epoxycholesterol on ALEXR, because we found this is an agonist. So I was wondering if that ligand or 24-25 epoxycholesterol could also be an ALEXR ligand in tumor cells that is being antagonized by the DDA.
Yes, sure. If you actually, what we found is that we can antagonize a number of effects induced by ALEXR ligand. So we haven't tested 25, 24 epoxide of cholesterol, but if it's an agonist on the receptor, yes, DDA would antagonize its effect.
And do you know if it is expressed in the tumor cells? I'm thinking whether they may represent as well an endogenous ligand that plays a role in tumors.
Actually, we haven't found DDA in tumors or at low level, and we know now that the enzyme is mostly absent from tumors.
Okay.
What could be possible also is that a number of supplementary transformation can be involved in the metabolic fate of DDA that could also produce a number of different effects that are not those induced by DDA. And this is also something we are working on.
And then I have a curiosity. I don't know if because of the way it works that you explained with histamine in the synthesis of DDA, I was wondering if you have thought of looking into histamine knockout animals so that you could actually see whether this is giving rise to more tumors in these animals or whether it is described at all.
This has been already described actually, but we haven't looked at that, but this is a good point. They also observed that the immune system was terrifically affected. But we know that histamine itself is very important to control inflammation. But it could be also that some of its metabolites, that like DDA, could be impaired in such mice and also affect a number of physiological processes related to immunity. Yes.
Yeah, it's very interesting because histamine it's also associated to depression, and depression is also associated to breast cancer.
Yes, that's a good point, Ernest.
Okay.
Thank you so much.
Thank you, Ernest.
Okay, next question from Magdalena Rudzinska. Magdalena?
Yes, it's me. Hello. I am food technologies from Poznań University of Life Sciences from Poland. And I work some years about the oxysterols. And I would like to ask you if you think that epoxides, derivatives of cholesterol, which are formed in food products and which are formed in human organism, they play the same role in our organism, in our blood. So what do you think about it?
This is a very interesting question, and actually this is something we are looking at also, because as you pointed out, 5,6 epoxide of cholesterol are among the most important in mass product of oxidation of cholesterol, and the level can be strongly increased by food processing also. And
Yeah.
we are checking whether or not the presence or the increase of 5,6 epoxide of cholesterol in the food could enhance could stimulate the development of tumors in mice. And why? Because if you consider that epoxide can cross the epithelial, the intestinal barrel, and then you can imagine that you can feed tumors with epoxide that can be metabolized and transformed into oncosterone, and then that could be enough to stimulate the development of tumors. So that's a very important point. And
yeah. So I have one question, if I can.
Yes.
What do you think about the addition of phytosterols in food products? Because in these functional foods, we have high level of phytosterols. And these products contain also high level of epoxides of phytosterols.
That's a very interesting question. And actually, we have just answered a call to get a grant to look at that, actually, because cholesterol and phytosterol have been reported to have opposite properties related to cancer development. And it could be that metabolism of phytosterol could be either different from that of cholesterol, or similar, but then we can imagine that metabolites of phytosterol could have different properties than those of cholesterol.
Okay.
So that's a very interesting point.
Yeah. Thank you for a very interesting lecture.
Thank you, Magdalena.
Bye.
Bye-bye.
Okay, the next question's from Alex Odemart. Alex?
Hi, Mark. Thanks a lot. It's a great study and great results. And actually, you have nicely shown that the OCDDO metabolite acts as an oncosterone. And I wonder whether the hydroxylated metabolite formed by 11-beta-HSD1 could also have protective functions by antagonizing receptor activity.
That's you mean if cholestantriol?
Yes.
This is a very good point. Actually, what we know is that this is unpublished results, is that this compound is a weak ligand of glucocorticoid receptor. So we haven't tested whether or not it could antagonize oncosterone, because we need to use high concentration of cholestantriol, which is cytotoxic for cells. So this is something we were not able to look at like that. So it could be, but we cannot see it experimentally.
Thanks a lot.
Thanks, Alex.
We have a follow-up question from Dieter Luchon.
Yes, Magdalena, I think, brought it on the point. It's an excellent playground for cholesterol to increase to plant steroids and other derivatives, and perhaps also Bill can answer this question. Now, if these children with SLO, if they form this 7-dehydroepoxycholesterol as a cell toxic compound, so far we always looked at the 7-dehydrocholesterol or plant steroids and zetosterolemia, but now we can increase this to all these different epoxy steroids. So could it be that SLO, it's not the 7-dehydrocholesterol, but the epoxy cholesterol of this compound is toxic?
That's a good point, Dieter. Actually, we found several cross interaction between the two metabolic pathways, the one that is involved in the oncosterone biosynthesis or activity, and the one involved in the DDA biosynthesis, for example. So what could be checked also is whether or not yes, because these but it's not only on SLO but also on NPC, that Newman-Pixie syndromes are associated with a high amount of cholesterol and 5,6 beta epoxide of cholesterol. So we think that these metabolites can interfere with key function driven by elixir or GR. And yes, according to their structure, it's because what we know is that a very small modification, the presence of a hydroxyl on one part of the compounds, affect completely the transcriptional response controlled by its receptor target. So that means that according to the structure of metabolite that accumulate, and now we need to explore in deep what kind of metabolite could accumulate on that kind of syndrome that could affect the physiology and be probably related, in addition to what we know related to the sonic H-Voc pathway, but that does not exclude, for example, the importance of elixir on such pathway also. So yeah, this is something that would be of interest to look at in the future.
Yeah, thanks.
Okay, so I have maybe the last question, unless there are any more coming. So Marc, what's your feeling with the formation of the epoxy? Do you think it's most likely to be dominated by an enzymatic reaction or via autooxidation?
I think that the two processes coexist. You know, it's clear that autooxidation is a major mechanism of epoxidation of cholesterol. Yes, it is known since a long time, but I think that the production of conjugates of 5,6 alpha epoxide of cholesterol might require the specific production of alpha epoxide of cholesterol. That means that either the metabolic fate of both isomers is different, and probably the gating to the enzyme, the target enzyme, will be different, or that there could exist also some interference between the metabolic pathway controlled by one or the other. And what I wanted to point it out, you know, that we found that oncosterone is a promoter or tumor promoter, like estradiol, for example. But that means that probably also a function of oncosterone would be also maybe to stimulate trophic effect or regenerative program in some tissues. That would explain also that it has been created for something and not for cancer development specifically. And I think that it could be some balance either during development or life between this pathway, because we have found some cross interference between oncosterone and DDA biosynthesis, and the reverse also, that could equilibrate that and maybe produce enough compounds to what is needed by the normal tissue. I don't know if I answer your question.
Yeah. So
and probably inflammation or lipid peroxidation could also perturbate that kind of pathway, and could be linked with inflammation-related disease such as cancer, but not only. can imagine that in neurodegenerative disease that could also occur.
Well, I think the nice thing is you've found these new mechanisms, and no doubt there's going to be lots more of the oxysterols for us to look at and to study their biological activity. So I think that this work has really opened up a whole new field for those of us who are interested in oxysterols to keep us going until at least we retire. So maybe there's one last question here in the questions. Okay.
And this is why we need collaborators also.
Yes. Okay, I think we've run out of questions. So thanks Marc for a really interesting talk. Thanks for asking all the questions, and thanks for everybody for joining the webinar and listening. Okay, I think I should tell you that before we finish that the next Lipid Maps webinar will be given by Stephen Blanksby from Australia, and that'll be in a month's time, I think. Okay, thanks everybody for coming, and thanks Marc for the lovely presentation. Okay.
Thank you, Bill.